We're going to take a look at Bacterial Flagella, Fimbriae, and Pili. Let's start with the Bacterial Flagella. They are made out of protein and easy to remember that their function is for movement. Not all bacteria have flagella, but some do. So this is important to know. This will definitely be on the test and the quiz. So make sure you know the patterns of Flagella distribution. So we've got peritrichous. So the word peritrichous, peri means around and trich means hair. And trich is just referring to what the flagella look like. So peritrichous just means that the bacterium has flagella all around the surface of the bacterium. And then the polar flagellar distribution pattern just means that there's flagella either at one end or both ends of the bacterial cell. And then there's some subcategories of polar, so they can be polar and monotrichous. Mono meaning one, so they just have one flagellum at one end of the bacterial cell. And then amphitrichous, remember that amphi means both, so they're going to have one flagellum, or they could have a tuft of flagellum at each end of the cell. So they're going to have one or a couple of flagellum at both ends of the bacterial cell. And then lophotrichous, lopho means tuft, and they're just going to have a tuft or a cluster of flagella at one end of the cell. So let's just take a look at some pictures. So the top right, we've got flagellum all the way around this bacillus, and so this one would be classified as peritrichous. And then over here on the top right, we've just got a flagellum at one end of the cell. So this one would be classified as polar and also monotrichous. And then right below that, we've got flagellum at both ends of the cell. So this one would be amphitrichous because amphi means both. And then over here on the bottom left, we've got a tuft or a cluster of flagella just at one end of the cell, and that one would be lophotrichous. So what I could do is I could put a picture like one of these on the quiz or the test and then ask what the flagellar distribution pattern is. So I definitely would know these. Okay, let's take a look at the parts of the flagellum. So we've got the filament, which is the long piece that extends out from the bacterial cell, and it's composed of a protein called flagellin. And this flagellin protein is not produced in our body, so our immune system will actually react to it and produce antibodies against it. So we've actually named the flagellin protein. Sometimes you'll hear it called the H antigen. And so one place that this is seen is there's a species of E. coli that causes disease in people called E. coli 0157 H7. And this actually comes from the intestines of cattle and can get usually in contaminated food. Especially if you don't cook your meat well enough. And sometimes it can get into contaminated water. But the H here is just referring to the flagellin protein found in this strain of E. coli. Okay, and then the next part of the flagellum is the hook that the filament will link into, and then the hook will then anchor into the basal body, and the basal body is what anchors the flagellum into the bacterial cell. So let's just take a look at some pictures here. So both of these are pretty much the same picture, but we've just got a gram-negative bacterial cell on the left and a gram-positive bacterial cell on the right. But you can see the basal body is going to anchor the flagellum into the bacterial cell. And then above that, we've got the hook. And then extending from the hook, we've got the long filament. So the bacterial flagellum rotate like a propeller. So they just spin in a circle, which you can actually see in this video here. They've actually glued the tip of the bacterial flagellum to the slide, so this bacterial cell in the middle can't go anywhere. And so you can just see how it just spins in a circle. And the way that the rotation goes, if it's rotating in a clockwise direction, which you don't need to memorize, then the bacteria will do what's called a run, which just means that the bacteria will swim in a fairly straight direction. But then when the bacterial flagellum stops and rotates in the opposite direction, counterclockwise, then we're going to do a tumble. So run and a tumble, those would be good words to know. And a tumble is just when the bacterial cell starts somersaulting, and then the flagellum will then stop and then rotate clockwise again. And then when it does another run again, it might take off in a different direction. And then it stops, rotates the opposite direction again, does another tumble, and then does another run. And so they will just do these series of runs, tumbles, runs, tumbles, runs, and that's how they swim around. Okay, so you don't need to know the clockwise, counterclockwise direction, but just know the terms run and tumble. So those are important to know. Okay, the next big thing to think about is what causes the flagellum to rotate. So I would definitely know that. So start with that in your notes. So looking at this picture here, here's a close-up of the basal body that actually spins to rotate the flagellum. Here's the hook and the filament. And anything in this picture that would provide energy to cause the flagellum to rotate and spin. So as you're looking at the picture, you might notice something familiar. So do you notice these hydrogen ions? Notice how they're slipping through this little channel here. And what these hydrogen ions are doing is they're going to be going down their concentration gradient. So we've got lots of hydrogen ions here and not as many inside the cell. And so the hydrogen ions are just going down their concentration gradient into the cell. And that's going to release energy and cause this basal body of the flagellum to rotate, and that'll cause the flagellum to rotate. So this hydrogen ion concentration gradient and those hydrogen ions moving, that's called the proton motive force. Because remember, hydrogen ions are the same thing as protons, and these protons or hydrogen ions are moving. And as they move, they generate a force, which will then, in this case, be used to move the flagellum. So that's going to be what's used to cause the flagellum to rotate, this proton motive force. And if you remember from the electron transport chain, the ATP synthase that spins around to shove the phosphate group onto ADP to make ATP, that also rotates because of the proton motive force as well. Okay, and then we also have this idea of chemotaxis. So these bacteria that have a flagellum and can swim around, they are able to move towards or away from things using these chemoreceptors, and they can move towards something good or move away from something that's bad. So in this picture, we've got up here on top just the random movement of a bacterium with a flagella just doing a run in a straight line. Here's where they've tumbled, and then they do another run, tumble, and you can just see they're just randomly moving around. But in the bottom picture, if we put some food over here on the right, you can see that they can move towards the food, and that's called chemotaxis. So they can use their chemoreceptors, these receptors that detect the food, and they can direct their movement, their swimming, towards the food. Now they can't really drive in a straight line towards the food, because they do a run and then they do a tumble, and then they're like, oops, kind of going the wrong direction. They do another tumble, straighten out again, they're like, oh, not quite right, we'll do another tumble, do another run, and so they eventually will make their way to the food, but it's not in a direct straight line, but they do make their way over there. So that's called chemotaxis. And then this picture over here on the left, the bacteria started growing in the middle, and as they ran out of food, they swam out where there was more food. And so you can see that they can swim towards good things. And so that would be one use of chemotaxis. They could swim out towards things they want. And then this picture over here on the right, they've got these little white discs where they put some nasty chemicals like vinegar, which is pretty gross. And they've got some different concentrations of vinegar. And what's happened is the bacteria are like, I don't want to be around the vinegar, and so they've swam away from the vinegar. And so you can see these clear areas around the disc that's soaked with the vinegar, and so the bacteria have swam away from gross things or bad things. So they can swim towards good things or swim away from bad things, and that's called chemotaxis. So they can direct their swimming, their movement. Let's take a look at the spirochetes next, and let's look at the video down here on the bottom left. These are some live spirochetes. You can see that they're thin and flexible. Notice how they're going around corners, bending, and here's a close-up view. And think about what type of microscope took this picture. We've got a dark background and a bright specimen. Hopefully you said it was a dark field microscope. Remember, the advantage of that type of microscope is that it increases contrast without staining. So you can look at these live microorganisms and see what they're doing while they're living without having to stain them and kill them. So these spirochetes have some different types of flagella. They're called axial filaments or endoflagella. Endo means inside or within. And their flagella are actually wrapped around the body of the bacterial cell. So they don't stick out at all. So they're in what we call the periplasmic space, which is the space between the cell wall and the outer membrane of the bacterial cell, and we'll explain the periplasmic space a little bit later. But when the endoflagellar axiofilaments start to rotate, they cause the spirochete bacteria to corkscrew through their environment. So we've got a couple of medically important spirochetes that I want you to know. So we'll put a star on these. So definitely know these for the test and the quiz. So a couple of examples here that I want you to know. First one is called Treponema pallidum. And this one causes the STD syphilis. So that is a spirochete. And our next spirochete example is called Borrelia burgdorferi. Oops. And that one causes Lyme disease. And that one's spread by tick bites. And then the last medically important spirochete that I want you to know is called Leptospira. And there's a number of different species of Leptospira bacteria, so we're not going to put a specific species. But the Leptospira bacteria cause a disease called leptospirosis. And how you get the Leptospira bacteria is you come into contact with urine from an infected mammal. And the Leptospira bacteria are often spread by rodents, but they can also be spread by a lot of other different mammals. So they could be spread by dogs and raccoons, possums, deer, cows. You know, there's a whole bunch of them. And so if you come into contact with the infected urine or soil that's been infected with the bacteria from the urine and it gets into a cut or into your mucous membranes, through maybe, you go swimming and, the water's contaminated or, gets into a cut or something, that's how you get infected. Leptospirosis, that disease, what the bacteria cause, depending on the species of bacteria that you get infected with, it could cause things like kidney failure, can even cause liver failure, meningitis, respiratory failures, so there's a lot of pretty serious things there. Luckily in Utah, we don't see too many cases of leptospirosis because we're so dry here. But other parts of the country, there are a lot more leptospirosis cases. Okay, so let's compare the bacterial and eukaryotic cell flagella because they are pretty different. So they are both made of proteins, but they're made out of different proteins. So the bacterial flagella are made out of a protein called flagellin. And then the eukaryotic flagella are made out of microtubules. And remember, those are made out of proteins as well. Remember, the microtubules are part of the cytoskeleton. Okay, the motion, the bacterial flagella, they spin around like a boat propeller. And then the eukaryotic flagella are more of a whip-like motion. And then the energy source to drive the movement. Remember the bacterial flagella, we talked about the proton motive force, that hydrogen ion concentration gradient. And eukaryotic flagella, they're going to use ATP as their energy source to move their flagella, so that's pretty different. And then the size, obviously bacteria are much smaller cells than eukaryotic cells, so the bacterial flagella you cannot see with a light microscope unless you stain them. So they're going to be much smaller, thinner, and so you can only see with the light microscope if you stain them. And you have to use some special stains which will stick to the flagella and make them thicker so you can actually see them. And then the eukaryotic flagella you can see with the light microscope because they're much bigger. Okay, the last two structures to talk about would be the fimbriae and pili. Fimbriae is plural and fimbria would be singular. And the fimbriae are made out of protein. They're very short, thin, and there are lots of them all over the bacterial cell. Not all bacteria have fimbriae, some do. And what the fimbriae are used for, which would be important to remember, is they're used to stick to surfaces. So that's what the fimbriae are used for. The pili, that's plural. Pilus would be the singular if you just had one of them. They're going to be much longer and thicker than the fimbriae, and usually you only have maybe one or just a few of them on the cell. And the uses, we're going to be looking at a sex pilus, which is going to be used in conjugation, which is where one bacterial cell will transfer DNA to a different bacterial cell. So that's what we'll look at. But some of the pili could be used for motility as well. So let's just look at some pictures. And here on the left, we've got an electron microscope view. The long structures are the flagella. And then all of these little hair-like structures, these are the fimbriae. So notice that they're very short, thin, and there's lots of them. And remember that these are going to be used to help the bacterial cell attach to things. So an example of a bacterial cell that has fimbriae would be E. coli. So when E. coli comes out of your GI tract and goes up your urethra, it gets into your bladder, how it avoids getting peed out and how it's so successful at causing UTIs is it uses its fimbriae to attach to the urethra and bladder wall, and then it doesn't get peed out. So that's how it can be very successful at causing a UTI. Okay, and then over here we've got two bacterial cells, and between them there is a sex pilus. So notice there's just one of them. It's much bigger than the fimbriae. And here we've got a piece of DNA that's getting ready to be passed from one bacterial cell to the other bacterial cell. And so this sex pilus is going to be used in something called conjugation, where one bacterial cell can pass a piece of DNA over to another bacterial cell. And so we'll learn more about that later.